And here we're going to look at excitation-contraction coupling. So this is the second important process for muscle contraction. This is what regulates muscle contraction. This is what turns it on and turns it off. So as you would imagine, the body being this highly efficient structures, right? This highly efficient system. We don't have muscles contracting indefinitely, and we don't have this process happening all the time. And so in order to regulate this process, calcium is going to play that role of turning contractions on, and then the absence of calcium is going to play the role of turning contractions off, okay? And so here we're going to speak about the sequence of events where an action potential, and if you remember from our last unit, we described this to be an end plate potential, where this action potential in the sarcolemma causes contraction. So how do we connect the dots from the events that happen on the neuromuscular junction where that end plate potential is brought about to the actual shortening of the sarcomere to the cross-bridge cycling? How do we fill in the steps of that communication? And that's what excitation-contraction coupling is. So this is going to depend on neural input at the neuromuscular junction coming from the motor neuron. That's going to result in release of our neurotransmitter, which is acetylcholine. And that requires calcium release from the sarcoplasmic reticulum, which is then important in interacting with the regulatory proteins on actin and turning on this muscle contraction, okay? So this slide is really a review of what we spoke about in terms of the events at the neuromuscular junction, the somatic nervous system. So we know that each motor neuron innervates several muscle fibers, and we also know that each muscle fiber receives neural input from only one motor neuron. And so this gave us the understanding of the motor unit, right? One motor neuron and all of the fibers that it was innervating. And so this is similar to an ordinary synapse in that acetylcholine is going to be released from the axon terminal, and that is going to bring about an end plate potential on the motor end plate. And if you remember, we described the motor end plate as being that highly folded membrane of the skeletal muscle fiber. And it's highly folded in order to increase the surface area, which will help to spread that end plate potential and transition or convert that electric energy, that end plate potential, throughout the entire muscle fiber and bring about contraction. Okay. So let's go on to speak about the role of calcium in all of this. So if you remember the two regulatory proteins, troponin and tropomyosin, that are interacting with actin, this is where calcium comes into the picture. So troponin is going to have a binding place for calcium. And if there is no calcium bound to troponin, tropomyosin is going to cover up the binding spots on actin and essentially prevent myosin from binding onto actin. Okay, I'll say that again. So if there's no calcium bound onto troponin, remember troponin has a binding spot for actin, for tropomyosin, as well as for calcium. So if there's no calcium on that calcium spot, then tropomyosin is going to cover up those binding spots, and we cannot have cross-bridge cycling because actin and myosin are not going to be able to interact. In this state, muscle is relaxed and we do not have contraction. Now, if calcium is present, it's going to bind onto that binding spot on troponin, and it's going to slide tropomyosin off of those actin, off of those myosin binding spots on actin, and that is going to uncover those binding spots and allow myosin to reach up and bind onto those binding spots, essentially forming that cross-bridge cycling, and in that state, muscle will begin to contract. So this is how calcium regulates this process. If there's no calcium, muscle contraction is turned off and the muscle is in its relaxed state. And in the presence of calcium, muscle contraction is turned on and the muscle is in its contracted state. So here are the steps that govern or bring about that excitation contraction coupling. And the first one is going to be the action potential generated in the sarcolemma. So that's really going to pick us up where we left off, describing the events of the neuromuscular junction. We bring that neurotransmitter across the cleft, it binds onto those receptors, those nicotinic receptors, and we generate an action potential, which we described as an end plate potential, and that is propagated throughout the muscle with the help of the T-tubules. So the T-tubules is how we propagate this action potential throughout the muscle. And then we've got these receptors that are on the sarcoplasmic reticulum that can couple the binding of calcium to that end plate potential. And so when we get that muscle when we get that end plate potential running through the T-tubules, that is going to bring about the opening up of the channels on the sarcoplasmic reticulum such that calcium can flood the cytosol of the muscle fiber. It can bind onto troponin. It can shift tropomyosin off of those binding sites, and then cross-bridge cycling can be initiated. Right? So these are the 6 steps, and we're actually going to walk through this schematic image that kind of depicts these steps in more detail. So the first step is acetylcholine is released from the axon terminal of the neuromuscular junction here. It's going to cross the cleft, and it's going to bind on to these nicotinic cholinergic receptors, and that is going to bring about an end plate potential at the motor end plate. This end plate potential, or this action potential, is going to propagate throughout the entire muscle body. It's going to travel along the T-tubules, which are these invaginations that we talked about. Remember, we described the triad as being the T-tubule in the center, and then the lateral sacs, which are formed by the sarcoplasmic reticulum. And so the T-tubules are these invaginations or these tunnels that help to spread the action potential throughout the muscle very quickly. The third step here is that the action potential is going to bind to receptors that are in the sarcoplasmic reticulum and trigger the release of calcium from the storage area, which is in the SR, out into the cytosol of the muscle fiber. So let's back up here a little bit. If we remember that the sarcoplasmic reticulum, which in muscle is the storage area for calcium. This is where calcium is being stored. And this is why we don't have muscle contracting all the time. The calcium that is important for regulating this process is actually stored away in the SR until the action potential turns these receptors on that actually open up these channels and bring this calcium out into the cytoplasm. Alrighty? And so when that calcium floods the cytoplasm, it now comes into contact with actin, specifically the binding site on troponin, which is this pink regulatory protein. Calcium can bind onto troponin, and then that can drag tropomyosin. So tropomyosin is this thread-like protein in purple. And once that gets dragged off of these myosin binding sites, then myosin can reach up and interact with these myosin binding sites on actin and start to bring about cross-bridge cycling. Now, after that is done, we must get rid of all the calcium that's in the cytosol. This is very important for ceasing muscle contraction. Otherwise, we would have muscle contracting indefinitely, which we do not want. So we must actively transport this calcium from the cytosol or the cytoplasm back into the sarcoplasmic reticulum, where it can be stored until another end plate potential or another action potential is brought about. And so once that happens, if there's no longer calcium present in the cytosol, then the troponin will not have calcium bound onto it. And so it's going to drag tropomyosin back to cover up those myosin binding sites once again. And that is going to bring about relaxation of the muscle as actin and myosin can no longer come in contact with each other. Okay. All righty. So here is what actin looks like in its two states. In its relaxed state, we can see that troponin, which is one of those regulatory proteins associated with actin, does not have calcium bound onto it. And I keep describing this relationship here. Remember, troponin has 3 binding spots. It has a spot to bind calcium. It has a spot to bind tropomyosin, which is this thread-like regulatory protein, and it has a spot to bind the actual actin filament. And so when it doesn't have calcium on here, it's going to drag that tropomyosin to cover up these myosin binding sites. And so in this relaxed state, actin cannot interact with myosin because these myosin binding spots are covered up. In its contracted state, actin does not have these tropomyosin filaments covering up the binding sites for myosin. And that's because calcium binds onto its spot on troponin, and that drags this tropomyosin protein off of the binding sites, uncovering them so that the myosin head can reach up and make contact at these exposed myosin binding sites. So this is how we say that calcium regulates this process. If there's no calcium, actin cannot bind to myosin. We cannot have cross-bridge cycling or shortening or contracting of the muscle. If there is calcium, actin is now exposed, right? Those myosin heads on actin are now exposed. And so myosin can interact with actin and bring about that cross-bridge cycling or shortening of the sarcomere. All right, so I'm going to pause here once again and see what questions may come up. So that is essentially the crux, the important parts of excitation, contraction, coupling. And again, there's another sort of short, concise video that will summarize these events. So these are the 2 important processes of understanding how muscle contraction is brought about. And just to summarize here, we talked about the steps of how the actual shortening comes about. And then we spoke about the steps of how this is regulated.